Thanks. All right. Hi, everybody. Good morning. My name is Melanie Sifton, and I'm a member of the Thomas and Smith Labs at University of Toronto in the Daniels Forestry Division. And I'm a PhD candidate with the focus on plant-soil interactions. And specifically, I'm testing the use of soil amendments to improve plant outcomes in difficult urban soils. My background is as a horticulturist. I'm actually a recovering botanic garden manager with a troubled past full of frustrations in trying to cultivate plants in the shadows of skyscrapers, run community gardens in contaminated soil, and manage street tree stewardship programs in roadside pits. So today I'm going to be presenting some work that I'm preparing for publication as part of my PhD thesis. So to start off, just want to go over some of the key urban soil challenges. As I mentioned, my focus for my PhD work is on constructed landscapes where soil topography and profiles are primarily engineered, and they're often very manipulated by humans, particularly when we are looking at creating new parks and typical home development sites. So the types of soils that I'm working on are primarily anthrosols, which are human disturbed or manipulated soils, and technosols, which are constructed soil-like media, usually used for growing plants in specific conditions, such as green roofs and nursery potting media. Over the long run, even very disturbed soils and even toxic landscapes can become more functional through pioneering species and adaptation to new conditions by organisms. But sometimes we don't want to wait for this, particularly in modern cities, and nor do we readily allow for it. So some studies, such as Sharonbrock in 2005, have showed that urban soils that have been disturbed and compacted by human development can have healthy levels of organic matter and properties of healthy undisturbed soil at their upper horizons after about 50 or more years. But what can we do to speed this up and to speed up their effectiveness in being able to grow trees and plants in these disturbed conditions? So with that overarching question, this particular project asked whether we can use nitrogen fixing clovers with biochar as a soil amendment to help trees grow better in disturbed urban soils. The use of biochar with nitrogen fixing species has been shown to enhance productivity of agricultural systems. So both nitrogen fixing crops as well as in mixed species systems have shown promise in this. So with that, we looked at the potential for use of biochar in combination with nitrogen fixing companion plants within an urban forestry context. So this was a 3-month factorial greenhouse experiment comparing growth responses in silver maples, which is Acer saccharinum. So we use saplings planted with 2 types of biochar, soil amendments, both alone and then in combination with 2 nitrogen fixing plant companions. So before I go too far, I think I better explain what biochar is. It's a charcoal-like soil amendment created by pyrolysis, which is decomposition of organic material at high temperature and in the absence of or in low oxygen environments. So in the case of this study and in all my studies for my PhD work, I'm working with Canadian wood byproducts, mostly sawdust and materials that are byproducts of the forestry industry. And I'm also generally looking at using biochars that are pyrolyzed at a relatively high temperature, so normally over 600 degrees Celsius. So the results of these generally produce what's called the high temperature wood biochar. And you also get a byproduct of biofuels in most cases as well. So biochar plants that are creating this as soil enhancement amendments also tend to produce biofuels as well. The Clover Companions that I worked with in this study are Trifolium repens, which is a Dutch white clover. It's a non-native species, but it's widely established in North America. It is a nitrogen fixer that's often found as a weed, but it's sometimes deliberately sown into lawns or meadows as a cover crop. The other nitrogen fixing clover is a native species, technically not a trifolium or clover, but it's called a prairie clover. The species is Dahlia purpurea, or purple prairie clover. It is native, but it's a lesser known species as a cover crop. Both of these species do have associations with nitrogen fixing bacteria, which is a rhizobium bacteria, which induces root nodulation and nitrogen fixation to support the plant. And often that is helpful as in pioneering environments where not much else is grown and nitrogen can be a challenge in disturbed soils. So the combination of biochar and plant growth promoting microbes, such as the rhizobium bacteria associated with these cover crops, can be used to reestablish urban vegetation, we believe, to help move disturbed urban soils more quickly towards functional restoration. So the proposal with this project is that these combinations can help increase nitrogen availability to a target species. Nitrogen is a primary plant nutrient, but a challenge with biochar, though it's been shown in general to reduce compaction in soil, to be able to bind toxic chemicals, to increase soil moisture availability, and to generally improve soil nutrient availability. It has sometimes been shown to bind nitrogen in the soil. And so with this study, we were trying to see if the nitrogen fixing cover crops and their rhizobial partners could be helpful in overcoming nitrogen binding issues that are sometimes found with some biochars. Now, few studies have looked at these combinations before. Even rarer are studies pairing herbaceous nitrogen fixing cover crops for support of trees and woody species. None of these have before looked at combination of biochars and low growing nitrogen fixing cover crops for urban forestry. So the hypothesis was that nitrogen fixing clovers could help increase the growth of maples with biochar. In particular, we expected that trifolium repens would induce the most growth response in the trees. So silver maples were 1-year-old bare roots planted in 1-gallon pots, and we introduced them to disturbed loam topsoil, which was stripped from a construction site in Whitby, Ontario. So the soil was combined with either sugar maple biochar or mixed conifer granulated biochar. Equal numbers of seeds of trifolium repens and Dahlia purpurea were sown to compare the non-native and native clovers as cover crops with and without biochar. So the argument behind this was that Thomas et al. In 2019 had showed that leukina and maize had an over-yielding effect with biochar and not without. So over-yielding is the increase in biomass in the mixed system over what is found with the crop in monoculture alone. Studies prior have looked at yield effects on 2 herbaceous crops, but this study was an attempt to see that if any over-yielding effects could be found with the herbaceous legumes paired with a woody crop, in this case, silver maple. Many other studies around the world have shown that the addition of biochar to soils growing bacterial nitrogen-fixing legumes will increase the nitrogen-fixing capacity of the rhizobia. So this has been shown over and over again, primarily in agricultural systems. One theory is that the rhizobia can readily colonize the biochar micropores and benefit from a likely reduction of oxygen in the tiny pore spaces, which helps to stimulate nitrogen fixation. But the mechanism behind this increase in nitrogen fixation with biochar is not completely known at this time. So here's a quick list of some of the data that I collected and analyzed, primarily using ANOVA and ANCOVA analyses. If anyone has questions about some of the equipment that I use for this, yeah, you can ask me later. I don't have time to go into all of it at this point. So quickly go into a few of the results. Why is it not advancing? I think somebody's trying to get into the room and I can't advance at this moment. Let's see. There. Okay. Nope, that went back. Okay, here we go. Forward. All right. So some results that were shown after the end of the 3-month study. So starting in the top left and moving clockwise, the highest tree growth performance was obtained with a combination of both biochar and nitrogen fixing plants, with up to a 30% increase in biomass compared to controls for the granulated biochar and a 55% increase for the sugar maple biochar. Particularly notable is the combination of sugar maple biochar and trifolium repens. So root fraction, which is the ratio of root mass to total biomass in the top right-hand corner, also showed that biochar in combination with the clovers showed some increases. Root mass increased over control when biochar and clovers were combined. But this analysis also showed that conifer biochar granules combined with the clovers was helping to build root mass compared to control. And significant was trifolium, particularly with conifer biochar. Final height analysis showed that biochar alone was helping to increase the height of the trees, while trifolium alone was not, either with or without biochar. And finally, in the lower right-hand corner, leaf area analysis strongly showed that sugar maple biochar alone, and even more so when combined with clovers, increased leaf area of the maples. Clover and biochar effects on soil pH showed that pH was higher in most of the treatments. This was a bit unexpected, particularly conifer wood biochar granules raised pH higher and more consistently than sugar maple biochar. We actually expected the opposite. Soil electroconductivity was increased strongly with conifer biochar granules, while it was reduced by sugar maple biochar. Maple water use efficiency was not shown to be significantly different than control between treatment groups, nor soil moisture. Neither of these were actually significant, but we can still see here in the figure on the right that there was some potential soil moisture reduction by trifolium treatments, the Dutch white clover, and that indicates a possible competitive effect of those particular clovers for water in this particular study for the trees. Now, leaf nitrogen was significantly higher in 4 treatments compared to control. The biggest jump was with the trifolium repens combined with sugar maple biochar. This is actually what we expected we would see in order to prove that nitrogen fixing companions can help biochar overcome its reputation for binding nitrogen. In this experiment, the nitrogen binding does not seem to be a problem, however, since nitrogen increased in the leaves with only biochar as well as in combinations with the clover. So in summary, both biochar types have mostly positive effects on tree growth. However, sugar maple feedstock biochar resulted in greater increases in tree performance relative to the granulated conifer biochar feedstock. The nitrogen fixing companion species also increased tree growth in both cases, although faster growing trifolium reduced soil moisture content. And so we conclude that biochar additions in combination with nitrogen fixing companion species have considerable promise in an urban forestry context, but that optimization of the system in terms of biochar type and species combination is an important consideration for future research and applications. So thank you very much. Thanks especially to Professor Sean Thomas, the Thomas Lab, Professor Sandy Smith, and the Bugs Lab, the Green Roof Innovation Technology Lab teams at the University of Toronto, Ontario Centers of Excellence, and the Ontario Graduate Scholars Program for research support.